This is a revision video on PCR. So there are 7 revision questions. Pause the video, attempt the questions, and then you can press play again and go through the answers. Okay, so let's review the answers. PCR stands for polymerase chain reaction, and the purpose, DNA amplification. Okay. Question 3. So the 5 essential ingredients. So we've got a tube here, we've got some buffer, and we're going to add in to that reaction buffer some key ingredients. So 1, you should have okay, and we will talk about the type of DNA polymerase used in the following question. And finally, the 5th ingredient, sort of not an ingredient, but more of a device, is PCR machine. So obviously close the lid and then place that tube in the PCR machine, program the PCR machine to carry out the reaction. So let's take a look at the stages of that reaction for question 4. So you begin with the process of separation. And that's obviously breaking hydrogen bonds between bases to separate the DNA into single strands. And then the next step, okay, so those short single-stranded fragments of DNA that are complementary to the ends of the DNA for amplification. So our primers are about 20 nucleotides long. And then the next stage. And so the DNA polymerase used is a polymerase from thermophilic bacteria, so bacteria that live at high temperature. So the polymerase is adapted to function optimally at high temperature, so can work at 72 degrees. And then back to separation again. So this process is a cycle, and normally it repeats around 30 times. Okay, so that's the stages of PCR. So on to question 5. So we've got 400 base pairs, and PCR polymerase works at 20 nucleotides per second, so it's simply 500 divided by 20, and that gives us 25 seconds. And then question 6. So we've got a total of 30 cycles. We know that separation is 30 seconds and annealing 10 seconds, and we know that synthesis is 25 seconds. So if we add those together, we know that one cycle is going to take 65 seconds. And then we're going to do that 30 times, so we need to multiply that by 30, and that gives us 1,950 seconds. And then we need to convert that into minutes. Divided by 60, that's 32.5 minutes, and obviously 0.5 of a minute is 30 seconds, so your answer should be 32 minutes, 30 seconds. So in reality, a PCR reaction takes longer than this, close to an hour, because of time to move between temperatures and also some initial separation step. Okay, on to question 7. So remember, this is a doubling. So for this, since it's a doubling, it's 2 to the power of the number of cycles. So that's 2 to the power 30, and that gives us this number. But then this is happening for each of the fragments, so we multiply that by 1,000. And so at the end of this reaction, from 1,000 fragments of DNA, we end up with 1.07 times 10 to the 12 DNA fragments. So you can see how this can amplify DNA from a small amount into a huge number of DNA fragments. Okay, so that's the revision video on PCR.